Okay, everyone, so it looks like it's time to get going with the immunology lecture. So, what you need to know from this lecture, just the basics of the immune system, which I think is really cool. It's probably my second favorite lecture for this course, just behind the metabolism lecture. It just seems like the immune system really does act like a machine to like stop you from getting infections all the time. So, I think it's an amazing system in this class. It's definitely not advanced immunology. I would highly recommend you guys take immunology if you have the time. I think it's like MM Bio 481 or something. Anyway, that is, I couldn't take it. I took the infection and immunity class, the MM Bio like 221, and that was a good class, but I don't really care about virology or bacteriology, which are the like the class is split into thirds. I just really like the immune system. So I wish I could have taken that immunology course, but it's fine. Um, so these are the things you guys need to know for this lecture. So what's innate, what's acquired immunity, the basics of what a pathogen is, because there's a lot of debate about what pathogens actually are, like what they have to do to be considered pathogens, and we don't get into that debate at all in this class. So what tissues are important for the immune system? You guys probably already know the lymph system. If so, you can skip that part of the lecture because we're not going more in depth than that. What is the complement cascade? So that's something that I had to learn in my infection immunity class because I don't feel like that was taught to me in this course. So I'm glad you guys get a brief introduction into it. It's super important for portion of your innate immunity. And then, oops, sorry. Last thing, just describe different mechanisms of antigen presentation to either helper or cytotoxic T cells. So yeah, so antigen presentation is a big thing to know. Innate immunity, like what's the difference between that and acquired? Like, could you list that out? And then, like, what is the complement cascade? So let's go. First thing, so what are pathogens? So bacteria, viruses, fungi, obvious, right? Protozoa, parasites, foreign bodies. So this can just be like um, necessarily not even alive, but just foreign bodies that your body doesn't recognize will elicit an immune response, and chemicals and pollen. So hopefully you guys know pathogens are stuff that doesn't usually come from yourself, actually never, because your body would recognize it as self and not actually create a response to it. And they all contain antigens, and antigens are just in this definition something that an antibody can like bind to, something that an antibody can recognize. And there's like on one antigen there are many epitopes. And an epitope is like a specific amino acid sequence. It doesn't even have to be amino acid, actually. It could actually be like a fat molecule, like a bacteria have what's called LPS. That's a fat molecule that our body can like recognize as being foreign. So it doesn't have to be amino acid, but an epitope is the specific portion of an antigen that the antibody actually binds onto. So antigen is just something that the body can like build, can recognize as foreign, and then build antibodies against. Easy. Okay, so this is viral structure. You're not really tested on this, but it's just nice to know, especially with like SARS-CoV-2 going around right now. So that's actually the virus name. COVID-19 is the like disease caused by SARS-CoV-2. So if you guys didn't know that, that's kind of cool. So SARS-CoV-2 actually looks a lot like this. This is specifically influenza, but influenza and SARS-CoV-2 look very similar. Really, they only have a couple main differences, and it's these surface proteins on influenza are called hemoglobin and neuraminidase. Not hemoglobin, sorry. Neuraminidase and hemagglutinin. That's my bad. And on coronavirus, it's called, these would be called spike proteins. So they just have a different amino acid sequence, but they do have a capsid, and that capsid is like a lipid bilayer, like what we have. But then they also have, oh, sorry, the envelope is the lipid bilayer. A capsid is made up of proteins, and it can actually have proteins with like the DNA, or specifically for SARS-CoV-2 or influenza, like the RNA wrapped around the proteins can be the capsid. And then these steps, Dr. B is just telling you, like, I think he's only giving you this slide because he's an influenza researcher and he like loves learning about influenza. Um, and M2 is also on the surface, but it's just such a small amount in influenza that we don't really talk about it. So it has to be endocytosed, then it goes to the lysosome, which we'll talk about in this lecture, and it gets acidified. That allows for the RNA to be released. You have a reverse transcriptase. That's not true. So reverse transcriptases are found in viruses like HIV, which actually convert their RNA back to DNA. But influenza doesn't have a DNA intermediate, and so it's actually called an um, RNA-dependent RNA polymerase, or RDRP. You don't need to know that. That's so in depth. So really, sorry, you guys. Influenza does not have a reverse transcriptase enzyme, though. What I would take from this: influenza is like a ball. Anything else is like kind of not expected for you to know. This isn't a virology lecture; it's immunology. So sorry for getting a little bit lost. That just bothers me. Okay, so lymph nodes. You guys, that's all I'm really going to get into. So like you have the lymph, you know that the lymph helps circulate and keep like osmolarity balances between the tissues and the blood vessels and whatnot. And what's also really important is the spleen. And then you have like different tissues like the GALT, the gut associated lymphoid tissue. All this stuff like carries pathogens around in the body to be presented. Like in the lymph nodes, there'll be a lot of B cells and T cells that can see all of the antigens coming through and then elicit a response to them. So you think, well, what like immune system, what do I tie that to? I would tie that to the lymphatic system. Like that's how it circulates throughout the body. And I'm not going to spend too much time on this either because it's not important, honestly. I'm sure there'll probably be a question or two on it. So know what the spleen is, know about blood flow to the spleen. There's red pulp versus white pulp. If you want to stop and like learn more about this, go ahead and pause the video. I don't care about this and it's not important. So I'm going to keep moving on. Okay, this is decently important. You're not like specifically tested on this in this class, but you will be in other classes. So I'm going to go over it very briefly. So these are like the different white cells I would have in mind. And there's even more than this. These are just like the major ones we like to talk about. So you have dendritic cells, lymphocytes, monocytes, and macrophages, eosinophils, neutrophils, basophils. So by this, what I would know is neutrophils are 50 to 70% of the white blood cells that you'll find circulating. The next most common would be like lymphocyte and plasma cells, which would be your B and T cells and all that other stuff as well. And then these are just named based on like their lineage, like how they delineate to become lymphocytes. They'll have like different precursors to get where they're going to be these different molecules. And they all have a little bit different of a response in the body. And you can see those down here. Like, so if you want, again, you can take a pause. Not the most important lecture, not the most important slide for what you guys are tested on. This is a very important slide for what you guys will be tested on though. So innate versus acquired immunity. So I want you to think of innate immunity as just being always present, always ready to go, no matter what. And acquired immunity is like an acquired taste, right? Like you have to have like your mom's lasagna a couple times, like maybe 10 times before you actually like it. Acquired immunity is more like you have to do all these things before your body mounts a response. So there's actually a lot of infections that can be cleared out of your body by just the innate immune system. And don't think of like we label them as separate as like scientists to like help us understand like how stuff is functioning, but it's not like they're acting independent of one another. They are tied together. Like the innate immune system activates the acquired immune system, and the acquired acquired immunity, acquired immune system then helps like they're kind of like it can act as cheerleaders for the innate immune system, like get them pumped up, get them revved up. So like they're very interconnected, but we do like primarily associate some things with the innate immune system and some things with the acquired immune system. This is tested on the MCAT. I don't know about the DAT, but you should know these things. So innate immune system, phagocytosis is one. And I think that's actually, yep, that's the next slide. So I'll get more into that in the next slide. Don't worry about it. Any inflammatory response that you have is actually going to be a part of most of the time a part of the innate immune system, because that's like, picture that you get a cut on your like forearm. Okay, so cut your forearm with your pencil. Don't actually do that, but like pretend cut your forearm with a pencil. All of a sudden now you have like these bacteria like flowing in and viruses and whatever, and there's already immune tissue out there. There's macrophages reside in the tissue, right? So they already see that infection before your B and T cells see it, which are your acquired immune system, which are found in your lymph nodes. So there's already a response occurring, and inflammation is so common with infection because inflammation is the way your body allows more blood, more white blood cells, more T cells to get to that area of infection. Makes it easier for them to travel there. Inflammation actually is caused by certain molecules, and those molecules act as like signaling receptors. So like how a T cell doesn't have eyes, right? But how does it sense its environment? How does it know when to leave the bloodstream and go into the tissue? Well, it knows based on like the inflammatory molecules, the cytokines, the complement system that's released. So that's like a huge general overview, but I would say innate immunity is going to be like inflammatory, or like we're born with this. That's another word for innate. Chemotaxins, that's pretty basic, right? Like some cytokines we just will have, like neutrophils, and so like neutrophils and macrophages. When I think of the innate immune system, those are the number two thing, like number one macrophage, number two neutrophil that come to mind. These all play a role in innate immunity, except for the plasma cells over here. All of these other homeboys play an integral role for that. So that's an immune system, the innate more. So like phagocytosis is really important. So what's not innate? Because there is so much that's innate, right? So what's not innate is anything having to do with B and T cell activation. So T cell programming, I think we get to talk about that a little bit, which is so cool. B cells releasing antibodies or being presented antigens. That's a very specific, that's an acquired response to a specific pathogen. Whereas you can think of the innate system working for all pathogens that come in. Non-specifically, the acquired immune system is for a specific pathogen that we can recognize. Like the specific strain of influenza virus that we had last year would be like a specific pathogen. So if you see B or T cells or antibodies, any of that kind of stuff, that's acquired immunity. Everything else is really like innate immunity, honestly, which is kind of cool. So humoral immunity and cell mediated, we'll get into that as well in a little bit. Okay, so direct versus opsonized phagocytosis. So one, what is phagocytosis? And you guys think phagocytosis is a form of endocytosis? Yes, true. It's a form of endocytosis specifically done by only a couple cells in the body though. So we have to recognize that it's different than like your epithelial cells can't do this. Well, that's not true. Okay, pretend that's true for this class. Your epithelial cells can't do this, but your macrophages do, and your neutrophils can also do it, and your dendritic cells can do it. So really, like the only cells in your body that are doing phagocytosis are your immune cells. So specifically, we call this a phagocyte. I would think of this usually being a macrophage. A macrophage are like the main phagocytes in our body. So a pathogen comes along, it binds to one of these receptors, that activates endocytosis or phagocytosis, right? And it gets brought in. Okay, that's one way it can happen. A better, more efficient way is you have a pathogen with polysaccharide capsule. So if you see polysaccharide capsule, you're probably thinking bacteria, right? We didn't go over that, but you know that like bacteria have a cell wall made of like this glycosidic bond. So like there's sugars and amino acids there. You actually get antibodies binding to this pathogen. So this right here is showing you an interaction between, okay, let me be careful. There's two things that could be going on here. So one, this just says an opsonin molecule, and the primary opsonin molecule, like opsonization, is just surrounding a pathogen with markers the body recognizes. So antibodies are the number one like opsonizers in the body. So if you have an antibody response, you are thinking, okay, this is acquired. But why is the acquired system still producing phagocytosis? Well, phagocytosis is such an amazing way to get rid of a lot of diseases. What happens is your B cells will actually produce antibodies that recognize this guy, and then your macrophages can recognize the constant region of your antibodies and then bring it in better, like more efficiently than just bringing in a random pathogen. It knows exactly what it's looking for, and it can bind it specifically. So if it's an antibody, then that would be like acquired immunity helping the innate immunity. But if it's like a complement protein, which can also be an opsonizer, they look different than this though, then that would just be innate and innate immunity. So just know what phagocytosis is here, usually done by macrophages, can be done by other cells like neutrophils. And what's really cool is that we're learning more and more that like pathogens can actually induce the cells to phagocytose them in. So cells that aren't ready to do phagocytosis, because it's not just bringing the cell, this little pathogen into the cell. We have to learn about what it's going to do next, which is lysosomal fusion. So some cells wouldn't be able to do lysosomal fusion, so the pathogen can get onto their surface, tell them to bring them in to phagocytose them, and then that's how they infect the cell. It's kind of cool. So what's next? So now you are a pathogen, you're brought in. So right here, you're on, so this is our macrophage. You were just phagocytosed, and now you're in this little blub, right, inside of the macrophage. What actually happens now is you have a lysosome, which has all these killer molecules that just would destroy us if they were ruptured, and they'll also destroy pathogens. So what happens is the lysosome fuses with the endosome. So this is called the endolysosome. And then all of a sudden this pathogen gets broken down. So what you have in here are like enzymes that break down proteins. You have what he's showing you here, like this is, oh, I can't think of the word. Hydrogen peroxide. So that's a reactive oxygen species, just an oxygen radical, reactive oxygen species. So reactive oxygen species, or ROS, you will see a lot when talking about like immunology. It actually comes up in metabolism as well, but they're used to break stuff down. And you may think like, well, why is the lysosome able to carry them? There's a specific lining in the lysosome our body makes, kind of like in the gut, you know? So like there's that really low, like really low pH. Our lysosomes have a way to protect themselves as well from all these crazy things. So that's the lysosome. Okay, complement cascade. This is part of the innate immune system. And it's saying C-reactive proteins up top here, because every protein, I don't know why they decided to do this to us, but every protein in the complement system is named C, like C5, C5a, C5b, C2, C1, C4, C like C20, C6, C11, C9, C8. Sorry, I was just listing all the ones I could remember from my class. You don't need to know any of those, by the way. Just know that they're all called, like, they're all C proteins, C for complement, and that's just, I guess, the best way we could possibly do that. So the complement cascade has a couple of really important things to know. The number one thing to know for this class is that they can recognize a pathogen by themselves. So this isn't acquired immunity. These little complement proteins are just floating around in your bloodstream at all times. So like if a pathogen gets into your blood, it can be killed by the complement system without like anything else. So this system's so cool, especially since I had no idea it even existed. But what happens is after a series of like enzymatic cleavages, meaning that like these proteins aren't active, right? So think about it. If they're just active in the bloodstream without a pathogen, you're screwed, because they're just going to tear apart your RBCs, your red blood cells, or your white blood cells. But they're not. So what happens is they recognize something on the pathogen surface that would be an antigen. And so what they do is they start cleaving each other. They start enzymatically like clipping each other, making each other active, really. And then they form these big pores inside the pathogen. So literally it's just like a huge channel, and you can see it here, like this channel. So what happens is through this channel, all of a sudden now everything is permeable to this pathogen. So you are like screwing up the osmolarity of this pathogen. You're literally just like shotgun blasting the surface of this guy, and all of a sudden all these like water and ions are flowing in down the gradients. Now they're not like regulated at all, and the pathogen bursts open and dies. Okay, so that's what the complement system does. That's like one of the main things it does. But if you do get to take the infection immunity or immunology course, you'll learn that there's so many other things that the complement cascade does too. But this is the one you need to know for the class. It specifically cleaves each other, like so with nothing else. All it needs is the pathogen and its buddies, cleaves each other, makes each other active, forms these massive holes. Everything can swell, breaks open the pathogen, pathogen dead. Okay? There's no like second messengers here. There's no like interaction with anything else. for what we're talking about, it can actually activate the acquired immune system, or it can bind to antibodies, or antibodies can activate it. But that's a little bit too much for this class. It's so interesting. So if you're interested, I go look up a Khan Academy video more, or talk to me after class because we can go over it if you want to. But just know big holes, osmolarity issues. Okay, humoral, that should be an O. Immunity, that just means like B cells, and B cells are named because they are born, they're born, they are created, and then they are also, they mature in the bone marrow. Whereas T cells are created in the bone marrow, but then they go and they mature in the thymus. So B cell versus T cell. So B cell, also known as like plasma cells, just kind of another name for it. There are like some differences between like just a naive B cell and before it can convert into a plasma cell. You don't need to know those differences for this class. Once again, you can look it up because it's interesting. But B cells are really cool because they are antibody producers, right? So let's start going over antibodies. So hopefully you guys have seen a picture of an antibody before. If not, that's totally fine. So this bottom portion is called the FC region. That's super important. So what this stands for is something constant. I have no idea what the F means. Maybe you guys can teach me, but this is the constant region of the antibody. And this region, that doesn't mean it's not different from antibody to antibody. As we can see here, there's multiple different kinds of antibodies that would have different FC regions. But what it does mean is that it's relatively conserved between antibody to antibody. So our cells can recognize this FC region. So remember when we were talking about phagocytosis and like the opsonizing molecule. So what would happen is you have up here the variable region, or the Fab region, that will bind to the pathogen, and then the phagosome, the macrophage, can recognize the FC region on one of its receptors, grab onto it, and bring the pathogen in. So this part recognized by our own body, this part grabs onto the pathogen, okay? So you do have two heavy chains here in purple. Both of those, oh, so sorry. Both of these are the heavy chains. The light chains are these little blue guys. Up here is the antigen binding site. So hopefully you guys can see that. And it's called, this is like the variable region. Like, so constant region is this bottom portion here. The variable region is this top portion here, and the hypervariable region is like the very tippity top. So that hypervariable region gives you like super specific response to pathogens. And these yellow guys are really important for some reason. Everyone likes to test us on this. I don't know why. I just feel like there's some things people like to test you on that aren't necessarily the most important things in the world, but you got to know them. These are disulfide bonds. So all of these chains are held together by cysteine residues. So yeah, I would know that though, disulfide bond. So this is an IgG. So this is the classic antibody that we picture in our mind. And it is actually one of the most, it's the most common antibody in the blood, I believe, but it's not the most common antibody we produce. I think the most common one that we produce is actually IgA, which is a dimer here. And it's so cool because of this like little clip thing you guys see here. This can survive in like the gut. And so it actually will attack pathogens in the gut. So if a pathogen gets past like epithelial tissue, gets down into your blood, the IgA will grab onto it and bring it back up into the gut. So cool. So there are monomers, IgD, IgE, and IgG. You guys do not need to know the differences between all these guys for this course. This is just a good introductory to like, if you do take any infection in the course, you will have to know these more in depth later on. Know that there are different kinds of antibodies. This one's really cool. IgM is the best opsonizing molecule, as it should make sense because one of these guys grabs onto the pathogen and look like, holy, this dude's massive, right? So he can really elicit that kind of response. Anyway, so IgD, IgE, IgA, IgM, IgG. So Ig, immunoglobulin subclass. And then there's even subclasses beyond this. So just know that there are antibodies and that they're different. What's really cool here is you have antibodies, excuse me. So say for instance, you're 2 years old and you get the influenza infection for the first time. So you're sick, And about 2 weeks after you actually get the infection, so time 0, get the infection, you peak at what antibodies you're gonna produce, and then it starts going down. But look here, what's really cool is it never goes down to 0, even weeks and weeks later. So that's because your B cells that produce your antibodies have like multiplied. They're just chilling out in the bone marrow, and they're actually secreting a very low level of the antibody, probably for the majority of the rest of your life. So very small levels, but it's there. So say you get infected again, right here, you're able to recognize that infection even before the week is over. At week 1, you have more antibody than you did at week 2, and then at week 2, you are like peaking. It's insane how many antibodies you have. So this is why we say like we really can't get the same infection more than once. It's really just that first time we can get it. And then like we can get so many more later on, so many more antibodies. Okay, this is the B cell clones. They just clone, clone, clone. That's boring. So I'm gonna move on. Primary, secondary response, we just talked about this. You have your B cells are presented antigen, start making antibodies, and then that takes a long time the first time, but secondary, they start making it super quickly, okay? This is all the different things that antibodies can do. You can stop and act, stop and look at this more. We've talked about all the important ones though. So cell-mediated immunity, this is what the thymus, so you think of T cell. You have 2 different types of T cells. You have helper T cells known as CD4 plus cells, and then CD8 cells are cytotoxic T cells. So antigen presentation is something important, but that's something else. So I'm gonna stay here for just a second. So your helper T cells are the ones you usually think of where they just, these helper T cells will just go and act like once they're presented antigen from a specific bacteria or like pathogen, they're activated and they'll go and help out the body in fighting off this infection. Um, and then cytotoxic T cells actually just kill the cells. So they are activated by different antigen presenters. So that's what we need to get into. So MHC1, this is present on all cells, so immune cells and non-immune cells. MHC2 is only present on antigen presenting cells, and you can see here, that's only macrophages, lymphocytes, and dendritic cells. So MHC1 presents itself. So say you're like a cytotoxic T cell, a killer T cell, like we like to call them. You're just going through scanning all the cells of your body at all given points in time. Like they're never inactive, and you're looking for the MHC1. Um, you're looking for the MHC1 receptors on those cells, and if you find one that's presenting antigen, like a viral or pathogenic antigen, you blow up that cell. You just kill it. Now, so MHC1 is going to be recognized more by like the cytotoxic T cells, and MHC2 will be recognized by the helper T cells. So if a killer T cell sees virus on an MHC2, it doesn't actually do anything. It just says, oh, okay, you got this under control, because the immune cells have phagocytosed up the antigen and present it on a different kind of MHC protein, which is, sorry, I should have said that it's the major histocompatibility complex. So it looks different on the surface, like what's presented on 2 looks different than what's presented on 1. So what you really need to know, it's kind of cool, it's in depth, but what I would take from this slide is both present foreign antigens. MHC1 also presents itself, like self antigen, so that the body knows, oh, this guy's a homie, like he belongs here. So MHC1 is self and antigen. MHC2 is just like antigen from your infection. Okay, so you get activated by like a T cell. We just started talking about this. So say that this is, let's see here, does it say specifically? No, it doesn't. So I'm going to call this a macrophage, and I'm going to call this MHC2. So if this is MHC2, I'm going to say this is a helper T cell that's recognizing it, because that's more traditional of what helper T cells recognize. Whereas if this was an MHC1, this would be more of a cytotoxic T cell. So we're just going to say MHC2, T cell then has a receptor that can associate with the antigen and say, oh yeah, I know that guy. Like I've seen that guy. or I've been created to like fight that guy. And then he gets activated through signal transduction, secondary like phosphorylation, secondary messengers, all that nonsense we talked about before in this course. Um, but yeah, it gets activated by antigen presentation. So this is how the innate system, macrophage or dendritic cell, talks to the acquired immune system, the T cell. So once the T cell is active, it starts kind of guiding the response. It's like the conductor. T cells are super, super cool. There's so much going on with T cells. You don't need to know any of this really. Like this isn't really covered on the test much, but go ahead, pause if you want to go over this. Pause if you want to go over this. These charts are amazingly helpful when you have to like actually know every single step for like the infection courses and know the cytokines that are talking to each other. You don't need to know any of that for this class. So don't need to know this either. So, but I would pause on each one of these slides and take a second because you guys know that Dr. B will probably ask you about these. But I'm just not going to go over them because they're not really that important to you. And we've talked about most of it already. Allergic reactions is kind of just a pathogen response. And Dr. B will go over this because it's super cool. I feel like I've already given you guys too much information for today, so I don't want to freak you out. How we recognize like different blood cells is also based on like antigen presentation and antibodies that we have. So like the immune system is so important in all of this nonsense, you guys. Okay, so let's see. This is it. Once again, like cytokines and stress, great. Like cytokines happen, they interact with immune cells, the brain, endocrine cells. So what this is showing, this is kind of like what Dr. B was talking about earlier in class. Primarily, we think of cytokines as being involved in the immune system, but they actually do have other roles as well in the body, which we don't really have to go over any of them. So before you guys log off, let me just like make sure you realize like what you do need to study and what you don't need to study. There's only like 4 questions on the immune system on the final. 3 actually, sorry. So what you need to make sure you understand is kind of what I hopefully highlighted more. So I know the difference between innate and acquired immunity. I would know what the complement system does, and I would also know like how antigen presentation works. If you don't understand antigen presentation, please ask questions during lecture. Or go look up a Khan Academy video because I'm sure they can teach it more in depth and better than I did. But just understand the basic differences between MHC1, MHC2, and then what like the different receptors are. Okay, you guys, so that's going to be it for the immune system. I hope you enjoyed that lecture. I think we only have 2 lectures left after this now. Right? No, we have 3. Oh shoot, I might have screwed something up. Okay, well, that's what we're going to stop doing today. Okay, I'll see you on Wednesday.